Hello, everyone, and a hearty welcome to this Genetic Engineering and Biotechnology News webinar. It's entitled CRISPRs, Ushering in a New Age of Gene Editing. CRISPRs, doesn't that word have a great ring to it? Now, people not involved in biotech might think CRISPRs are something from Kellogg's. But for all of you who work in the life sciences, you know CRISPRs have become one of the hottest technologies around. And why? Well, as the title of our webinar states, they're ushering in a new age of gene editing. To tell us more about CRISPRs and where the technology might take us, we've assembled a star panel of CRISPR experts. Let's meet them. Dr. Radolphe Barrangon serves as adjunct professor of food science at North Carolina State University. Dr. Barrangon will explore CRISPR-Cas systems and their applications. Dr. Feng Zhang carries out research at the McGovern Institute for Brain Research at MIT. He's an assistant professor in the Department of Brain and Cognitive Sciences. Dr. Zhang will present additional details on genome editing using CRISPR-Cas9 and also provide some examples of the system in action. Dr. George Church is professor of genetics at the Harvard Medical School. He's also director of personalgenomes.org, which provides the world's only open access information on human genomic, environmental, and trait data. Dr. Church is going to talk about CRISPR applications beyond tools, libraries, and disease models. I'm John Sterling, editor-in-chief of GEN, and I'm going to serve as moderator. After the panelists make their presentations, we'll hold a question and answer session. Feel free to send in a question for our panelists at any time during the webinar. Type your question into the Ask a Question box on the lower left of your console and then hit Submit. The panel will try to answer as many as possible. So if everyone's ready, let's get going. Radolphe Barrangon will be our lead presenter, so I'm turning the program over to him. Radolphe?
Good afternoon. My primary objective today is to introduce CRISPR-Cas systems and provide an overview of their applications with focus on type 2 systems and specifically Cas9-mediated DNA cleavage and genome editing. The primary biological function in vivo of native CRISPR-Cas systems is actually to provide sequence-specific interfering RNA-mediated adaptive immunity in prokaryotes. These systems occur in nearly half of bacterial genome and the large majority of archaea as well. CRISPR-Cas systems are composed of two main elements. On one hand, the top right, the clustered, regularly interspaced short palindromic repeats, CRISPR, which form peculiar arrays of DNA repeats interspersed by elements called spacers. On the other hand, the top left, CRISPR-associated sequences, or Cas genes, which encode accessory proteins that, together with the CRISPR array, constitute what is called a CRISPR-Cas system. These systems do provide adaptive immunity in bacteria and archaea through three distinct molecular steps. The first step, acquisition, represents the vaccination stage during which a new spacer is spliced from an invasive exogenous nucleic acid, such as a phage or a plasmid, and this protospacer sequence yields a spatial sequence which is associated with the new CRISPR repeats, and this repeat spacer unit is specifically integrated at one end of the CRISPR locus, generating an immunized event. Secondly, during the expression stage, or the CRISPR RNA biogenesis step, the CRISPR array is transcribed as a long precursor CRISPR RNA pre-cRNA, which is subsequently processed into mature small interfering RNAs that typically contain a portion of a spacer and a portion of a CRISPR repeat. Lastly, during the interference stage, cRNAs, CRISPR RNAs, guide Cas endonucleases towards complementary nucleic acids for sequence-specific DNA cleavage. Altogether, those CRISPR-Cas systems provide adaptive immunity in bacteria and archaea. Now, overall, there are three distinct types of CRISPR-Cas systems, namely type 1, type 2, and type 3, and those systems carry distinct sets of Cas genes and corresponding Cas proteins as established by Makarova and Kunin in 2011. Cas1 and Cas2 are universally distributed, whereas Cas3, Cas9, and Cas10 are the signature genes that exclusively occur in types 1, 2, and 3, respectively. Noteworthy for today, Cas9 is actually the signature gene for type 2 systems, which happen to be the most streamlined CRISPR-Cas systems, as you can see here, with only 4 genes and proteins. Although the molecular underpinnings of these CRISPR-Cas systems vary between those 3 major types, there are many common mechanistic and biochemical processes that they share. Noteworthy, all 3 types rely on small interfering CRISPR RNAs, cRNAs, that all guide Cas nucleases, which eventually yield sequence-specific targeting and nucleic acid cleavage. For type 1 and type 2 systems, the system is specific for what's called a protospacer adjacent motif, protospacer associated motif, a PAM, on the cognate targets, and those elements are very important for target recognition and subsequent cleavage as well. Although most CRISPR-Cas systems target DNA, and you can notice on the bottom of the right that type 3B systems target RNA, but those are the exception rather than the rule. And interestingly, type 2 systems have thus far been exclusively observed in bacteria only. Now, if one looks at the CRISPR literature over time, it actually dates back all the way to 1987, where CRISPR arrays were first observed in an intergenic region flanking the IAP gene in E. coli. And these peculiar, unusual genetic loci were mostly anecdotal until they were repeatedly observed in the genomes of bacteria and archaea as microbial genome sequencing became widespread in the late 90s and early 2000s. And the CRISPR acronym itself was only coined in 2002, nearly 15 years after their initial observation in the E. coli sequence. Although they were originally and erroneously predicted to be involved in chromosome partitioning, transcriptional regulation, DNA repair, and other potential functions early on, the observation in 2005, actually 3 distinct papers published within a short period in the summer of 2005, all showed homology of spacers to viral and plasmid sequences in bacteria and archaea. And this led to the hypothesis within a year in 2006 postulated by Makarova and Kunin that those CRISPR-Cas systems may actually constitute an immune system in archaea and bacteria. Shortly thereafter, we actually showed in 2007 that indeed those CRISPR arrays in combination with Cas genes do provide adaptive immunity against viruses. And several subsequent studies in 2008 established that the overall process is RNA-mediated. That was shown by the Van der Roos lab from Wageningen University. And then within a couple of months, Sontheimer and Marafini showed that DNA is the primary target of CRISPR-Cas systems. We also showed in 2010 that CRISPR-Cas systems specifically cleave DNA. And then shortly thereafter, in 2011, 2012, and 2013, a few critical milestone discoveries established CRISPR-Cas systems for genome editing, which led us to the CRISPR craze. So if you look at the literature profile over time, it is rather obvious that the field has grown exponentially since 2007, and you can see firsthand on the right that the genome editing application that we've all witnessed in the last 12 months with the CRISPR craze in 2013 has led to an explosion of the CRISPR literature, CRISPR interest, and CRISPR applications. So if we look at CRISPR applications overall, there are 3 main types of CRISPR-based exploitations. Firstly, and originally, because spacers are acquired in a polarized manner at one end of the CRISPR locus, these peculiar genetic elements provide a genetic tape record of immunization events over time. They can accordingly be used to provide insights into the origin and the evolution of a strain or several strains, and be used to compare and contrast strains. This has been extensively used for bacterial phylogeny and epidemiological studies of foods and pathogen outbreaks, notably for E. coli and Salmonella. And this loci can also provide insights into host virus population dynamics and microbial diversity in complex samples and metagenomic surveys. A subsequent application has been the exploitation of their natural immune function to actually vaccinate strains of interest against exogenous nucleic acids like viruses. CRISPRs have been used already and exploited in the food industry for the natural development and formulation of dairy starter cultures that are commonly used, even as of today, for the global manufacturing of cheese and yogurt fermentation as well, where actually phage predation has been and still remains an industrial problem. We also showed that CRISPR-Cas systems can be leveraged for either natural or engineered vaccination against plasmids, and this can be exploited to prevent the uptake and dissemination of things like antibiotic-resistant genes or pathogenic traits. More recently, we showed that CRISPR-Cas systems can be engineered for self-targeting and may actually show some potential as next-generation antibiotics for strain-specific removal of selected bacterial species of strains, and there's some potential there that is yet to be unraveled. Lastly, and importantly for today, the molecular potential of Cas proteins, specifically Cas9, has already been harnessed and repurposed for a number of important applications, notably genome editing, transcriptional control, and genetic screens. So let's dig into Cas9 genetics, Cas9 functionality, and Cas9 biology. So we showed originally in 2007 that CRISPR spacers specifically provide adaptive and sequence-specific immunity against viruses and phages. Noteworthy, we showed that immunity can be reprogrammed by specifically engineering desired CRISPR spacers into a wild-type CRISPR array. And we also showed in that same study that Cas9, which was originally named Cas1, has also been named Cas5, was named CSN1 for a long time, and eventually Cas9. So Cas9 is necessary for CRISPR-dependent targeting, and spacers alone do not suffice for interference. We also showed a few years later, back in 2010, that Cas9 is actually involved in and responsible for plasmid DNA cleavage as well as phage DNA cleavage. And specifically, we showed in this study that blunt double-stranded DNA cleavage occurs exactly 3 nucleotides away from the 3' end of the protospacer sequence in the immediate vicinity of the PAM. This was done all in stress Haemophilus CRISPR-Cas systems. The same system was subsequently studied, and we also showed in 2011 that this native CRISPR-Cas system can be transferred heterogeneously to other bacteria, in this particular case from stress Haemophilus to E. coli, and can be readily reprogrammed to target plasmids or viruses, like in this particular case, phage lambda. We confirmed in this study as well that targeting strictly requires a Cas9, CSN1 on this slide, and we showed that the HNH motif was necessary for targeting and preventing the uptake and the acquisition of plasmid sequences by the strain. This same study also showed that targeting is heavily sequence specific, and actually that a perfect match at the 3' end of the spacer is necessary for cleavage, whereas in contrast, SNPs can be tolerated if they are located more than 5 or 5-ish nucleotides away from the cleavage sites at the 5' end of the protospacer. In contrast, a single SNP R or near the cleavage sites actually will prevent Cas9 endonucleolytic activity. And the same was shown for mutations in the PAM on the right, where you see that changing the GGNG PAM will preclude the occurrence of cleavage by Cas9. Lastly, we showed in 2012 that indeed there are 2 distinct important motifs for Cas9 cleavage namely RUVC and HNH, and that both are involved in the target DNA cleavage, and that actually each single motif serves as a DNA strand nickase, RUVC for the plus strand, and the HNH for the minus strand. So all those elements actually were scientifically interesting, but not as exciting to the global community until what I call the sgRNA tipping point. And in my opinion, the study by Jinek and colleagues at the Dana Lab at UC Berkeley did provide a tipping point, or the tipping point for the CRISPR field back in 2012, when they actually showed that the native CRISPR RNA tracer RNA duplex can be artificially replaced by a single guide RNA chimera, sgRNA, which links CRISPR RNA on one side and tracer RNA on the other side. And this really allowed this technology to change and be readily exploitable for flexible reprogramming and packaging of CRISPR-Cas systems using a single protein, namely Cas9, and a single molecule, namely the single guide RNA, for programmable and transferable DNA cleavage. So the CRISPR-Cas9 craze, or the CRISPR-Cas9 revolution, occurred shortly thereafter, and actually within a couple of months of the Jinek et al. Study, the Feng Zhang group, the George Church group, and the Marafini lab all concurrently and very elegantly and compellingly showed that this system, CRISPR-Cas9, can be readily packaged for heterologous genome editing, and those milestone studies arguably set the stage for the CRISPR-Cas9 genome editing craze we all witnessed in 2013, with arguably very significant translational and research impacts. So as we can see here, there are 4 distinct forms of Cas9 that can be readily exploited for various applications, one being the wild-type Cas9, which can be used to generate double-stranded breaks, whereas either an HNH minus nickase or a RUVC minus nickase can be used for single-strand DNA nicking. And lastly, an inactivated version of Cas9, widely called dCas9, which is inactive enzymatically but can solely bind DNA in a sequence-specific manner as shown in the Stanley Key lab. So those elements are, and will continue to be exploited for genome editing applications using CRISPR-Cas9, and essentially Cas9-mediated double-stranded break cleavage can be harnessed for targeted mutagenesis in combination with the native DNA repair machinery in various organisms. So as shown at the top, one option is the error-prone non-homologous end joining and HEJ repair, which can be used to generate either SNPs or insertions or deletions at the site of cleavage. Alternatively, another option is homologous recombination HR, which can be exploited for more surgical edits by specifically providing a DNA repair template with the engineered or desirable edits, which can be a SNP or could be an insertion or could be a deletion as well. Although the strep pyogenes model has been heavily used for the single guide RNA technology, there are other systems that can be readily exploited already, notably the Strathomorpholus systems I mentioned earlier today, and those can be used with the wild-type reduced spacer array as well. And there is a number of other Cas9 proteins in combination with their respective PAMs that have been and likely are being developed as well, perhaps even as we speak. So Cas9-mediated genome editing can be exploited in 2 different ways, either using the wild-type CRISPR system, which exploits the native CRISPR array in combination with Cas9, as well as the tracer RNA and RNase-3/4 processing, or alternatively, this system can be readily repackaged using exclusively Cas9 in combination with the sgRNA single guide RNA chimera. And the advantages of this technology include flexible, programmable targeting or reprogrammable targeting, sequence-specific DNA cleavage. This system can be readily heterogeneously transferred using codon-optimized versions or even NLS-containing versions of Cas9. The cleavage is arguably efficient, precise. The design overall is affordable, quick, and cloning and implementation can occur within days, if not weeks. And then the ability to multiplex the system using multiple guides concurrently or multiple Y-type spacers within an array is really promising for multiple edits, either iteratively or concurrently. And then last but not least, it was shown by the Church Lab that there are several distinct orthogonal systems that could potentially be used and paired together for different purposes. Now, nevertheless, notwithstanding the tremendous list of advantages that we see here, there are a few caveats to have in mind, notably the fact that Cas9 has and still remains a fairly large protein at above 4 kb and sometimes in excess of 1,400 amino acids. To a lesser extent, targeting precision is based on PAM sequences and availability. So for guide design or cleavage targeting, there's some flexibility, but it's not absolute whatsoever. And then last but not least, some groups have reported some off-target issues, but there are ways to alleviate those with optimal guide design, perhaps using longer PAMs. And as we'll hear later today, I think the elegant use of dual nicases for two nicks on different strands. So, in addition to Cas9-based genome editing, there are already other repurposing applications that have been established based on Cas9, notably the ability to exploit inactivated versions of Cas9 to repress transcription by blocking RNA polymerase, which has been coined CRISPR interference, CRISPR-I. And I think we are already looking at applications beyond genome editing. There are several recent developments that have shown you can use Cas9 and fuse it with different functional domains of interest for transcriptional activation, CRISPR-on, and possibly a whole new series of fusion proteins to manipulate DNA structure, DNA content, and even DNA function. So I think this here is the point in time at which we can already start anticipating the next generation of CRISPR applications. So at this point in time, I think we have a good understanding of CRISPR-Cas systems, what they are, how they work, and how specifically Cas9 proteins can be exploited for a number of applications, notably genome engineering and gene editing. And I would like to take a minute to acknowledge all my CRISPR collaborators over the years, notably my former group at DuPont, the Moino lab and the Cisnis lab in Vilnius with whom I've worked and presented some of their work today. I think this sets the stage for the subsequent talk we're going to hear from Feng Zhang and George. Back to John.
Thank you, Rodolphe, for that excellent overview of CRISPR's as genome editing tools. You've laid out the groundwork for the presentations to follow very well, so thanks again. If you are just now joining our webinar, welcome. As I mentioned earlier, we'll be conducting a Q&A segment following the panelists' presentations. Please type your question for any or all of our panelists into the Ask a Question box on the lower left of your console and then hit Submit. Our second panelist, Feng Zhang, is ready to begin his presentation. So, Feng, the floor is yours.
Hi, my name is Feng Zhang, and I'm an assistant professor at MIT and also a core member at the Broad Institute. CRISPR-Cas technology has started to enable a lot of very exciting applications in a variety of genetic and engineering experiments. One of the most exciting advances in the past decade has been the growth in our ability to analyze genetic variations in large numbers of individuals. So this is a plot from the National Institute of Health, and it shows the exponential growth in the number of publications reporting genome-wide association hits. One thing to remember from this plot is that these genetic variations are correlated or linked to a particular disease, but we actually don't know if they play a causal role. And so given this rapid rise, we also need a complementary technology to help us in a high throughput fashion, identify those genetic mutations that are causal in specific disease mechanisms. One of the approaches for quickly understanding the causal role of genetic variation is through genome engineering. The idea is very simple. You can start from a well-studied healthy stem cell, and by engineering the genome of this cell, you can then introduce a genetic variation that you would like to study and see whether or not it causes a disease state. And using this engineered cell, you can make a perfect comparison to the healthy stem cell because they only differ in the genetic mutation of interest. And by taking both of these cells, scientists can differentiate them into the relevant type of tissue. So if we want to study a neurological disease, we can convert them into neurons and then start to compare their morphology or physiology or any other type of property within the cells. So the differences observed can be attributed to the genetic mutation. And this is a technology that we can start to scale up and generate and study many different kinds of genetic variations at once. So how do you go about making a precise change within the genome to recapitulate a genetic mutation? It turns out that there is an elegant way to harness the endogenous machinery within the cell, either the non-homologous injoining pathway or the homology-directed repair pathway to make a change. This was first demonstrated by Dana Carroll and colleagues from the University of Utah back in 2002. And what we need to do to make this work is to introduce a double-strand break within the targeted region in the genome. And NHEJ allows us to knock out gene functions very efficiently because the double-strand break is rejoined with the formation of a small indel mutation. So if this indel is introduced in the coding region for a given gene, then it will lead to a frameshift mutation and then deplete the protein expression within the cell. Alternatively, the homology-directed repair pathway allows us to make a more precise change by providing a template that is homologous to the broken DNA ends, but carrying a small change that we want to introduce. It is possible to increase the efficiency through which the mutation can be incorporated into the target genome. So both of these technologies are possible to allow us to study the roles of genes and genetic variations, but what we need to do to make this happen efficiently is to create a double-strand break with high efficiency. To develop a way to make double-strand break efficiently, we turn to the CRISPR system. In addition to the CRISPR system, there has been multiple technologies developed over the past decade or two, including the zinc finger nuclease, the meganuclease, and also the transcription activator-like effector nuclease technology. But those technologies depend on the ability to engineer a protein to recognize a specific DNA sequence. What is elegant about CRISPR is that the specificity of DNA recognition is achieved through a short RNA sequence. And so by simply altering the composition of this RNA using Watson-Crick nucleic acid base pairing code, we can very easily target the enzyme to a specific location on the target genome. The CRISPR system is really one of the more elegant basic science stories. The system, as Rudolph told us, it was discovered in 1987, and then it wasn't until 20 years later that scientists have started to uncover the biological function. And then over the past 8 to 10 years, scientists have started to unravel the different kinds of mechanisms, starting from understanding the role of the system in antiviral defense to characterizing the systems in 2010 and 2011, the different enzyme and also RNA components that are necessary to carry out CRISPR activity. So when I started my lab in 2011, we turned to the system and thought, can we develop it for doing genome engineering in mammalian eukaryotic cells? So 3 graduate students joined my lab, Le Tong, An Ren, and also Patrick Su, and we set out to engineer 2 different kinds of CRISPR loci, one from the Streptococcus thermophilus bacteria, which is an organism that's typically used in the dairy industry, and also a pathogen from the Streptococcus pyogenes system. And both of these systems share very common features. They all have an enzyme called Cas9, And they also carry two non-coding RNA elements, one called the tracer RNA and another called the CRISPR repeat. The CRISPR repeat is where the variable sequences from phages are inserted into the bacterial genome. So based on the work from other groups that characterize the individual components of the CRISPR system, we thought to see, can we introduce the minimal but functional set of CRISPR elements into mammalian cells? And there were four different components that were essential. One is the Cas9 enzyme, which is the nucleus carrying out the cleavage activity, as Sylvain Moinu showed back in 2010. But also the two RNA elements, one for CRISPR repeat as well as for tracer RNA. So Emmanuel Charpentier also showed in 2011 that the RNase-3 from the host bacteria is required to mature the RNA into a form that can be loaded onto Cas9. So we also introduced the fourth component into cells. To test whether or not the system can modify the genomic DNA within a mammalian cell, we took the human genome and then re-engineered the CRISPR spacer sequence to target a specific 30 base pair sequence on the human genome in the EMX1 locus. And by introducing all four components and then making sure and engineering the tracer RNA so that it can express at sufficient levels, we found that introduction of all four components can indeed mediate double-strand breaks within the target locus in EMX1. And what is also interesting from one of our experiments is that you only need three, and you don't need the RNase-3 molecule. So likely there is a component within the mammalian cell that provides for that function as well. So while we were doing this work, other groups have continued to characterize the biochemistry and the biology of the system. And so two studies came out in 2012, one from the group of Emmanuel Charpentier and Jennifer Doudna, and another one from Virginia Sickness. And they demonstrate that Cas9 can indeed cleave DNA in vitro. What is interesting, and also as Radolphe pointed out earlier, is that the two RNA from CRISPR can be fused to form a single guide RNA. So we saw this and thought, can we compare the efficiency of the two different RNA forms? And we compared the single guide RNA reported in Genek et al. With the two RNA forms that we were introducing into cells. To our surprise, the single guide RNA actually didn't work very well. It was able to cleave in some cases, but the double RNA was by far much more robust. So we were a bit puzzled, and we started to compare the two different RNA forms. And we realized that one of the differences between the two RNA complex versus the single guide RNA is that the 3' region of the tracer RNA is much longer. There are secondary structures that are likely important for maintaining the stability or maintaining the interaction with the Cas9 protein. And so we thought to try to optimize this guide RNA. And one thing we did is we started to extend the 3' tail of the guide RNA. And what we found is as soon as we started to include one or both of the hairpins on the 3' region, we were able to significantly increase the cleavage activity of Cas9 within cells. And by doing a northern blot, we also found that these modifications further increase the presence, the level of expression of the guide RNA within cells. So George Church published a paper at the same time as our initial science study, and they also report the long form of the tracer of the single guide RNA to work most effectively. So that sums up the system. There are only two components that you need to introduce into cells. The first one is the Cas9 enzyme, and then the second one is the appropriately programmed guide RNA that targets the specific region of the genome that you want to cut. Once you do that, you can induce a double-strand break quite efficiently and then start to stimulate genome editing via either the non-homologous injoining or the homology-directed repair pathways. So here's one application of the CRISPR system to introduce a small mutation within the human genome. In this example, we use Cas9 to target and make a double-strand break in the EMX1 locus. And at the same time, we provided a single-stranded oligonucleotide template that is mostly homologous to the cleaved region, but in between the two homology arms, there is a small mutation. And in HTK cells, we found that CRISPR can induce up to 27% homologous recombination from the single-strand oligo. And in ES cells, we can get up to 6% homologous recombination. So both of these efficiencies are several orders of magnitude than the basal level of homologous recombination without introducing a double-strand break using CRISPR-Cas9. In addition to modifying cells, which are useful for generating cellular models, we worked with Radolphe at the Whitehead Institute, and we found that you can use CRISPR-Cas9 to engineer animal models with specific mutations efficiently as well. By taking single-cell fertilized embryos and injecting the messenger RNA for Cas9, as well as the guide RNA to target both the TET1 and also TET2 gene, we found that you can simultaneously knock out both genes, all four alleles, with up to 30% efficiency. Although sometimes in some animals there are mosaicism, this overall increases the throughput by which we can engineer animals with specific mutations. One of the important criteria for Cas9 function is that it has to be specific. So in one study, we took a single guide RNA and then computationally predicted all of the genomic regions that share high levels of similarity. So we identified a list of sites that differ by 1, 2, or 3 bases. What we found is that although Cas9 is unable to cleave at most of those sites, occasionally it is able to mediate a cleavage event at some of these sites that share high levels of similarity. What that means is we need to develop other ways to overcome the off-targeting concern of the Cas9 system. And so one strategy that we developed is by learning a lesson from the Talon or the zinc finger nuclease system, where rather than using a single guide RNA to recognize a single stretch of DNA sequence, we try to use 2 different guide RNAs so that we can double the number of bases that need to be recognized. And the way that this works is by first converting the wild-type Cas9, which can cleave both strands of DNA, into a nicking enzyme by mutating one of the 2 catalytic nuclease domains. And this system can work quite well. So as long as you follow a couple of criteria, for the D10A Cas9 nickase, where we inactivate the RUBC domain, we found that as long as the 2 guide RNAs are spaced not too far or not too close, with an offset of between 0 to 30 base pairs, and leaving a 5' overhang, then we can introduce indel mutations via NHEJ with similar levels of efficiency as using wild-type Cas9. And furthermore, this system is able to reduce off-target activity. So taking one of the guides, target 1, that we know to have 5 different off-target sites, we found that by switching to the double nicking system using the D10A nickase and 2 guides, target 1 and also target 9, we can overcome the NHEJ modification at the 5 known off-target sites. So this was able to increase the specificity by over 1,000-fold. So we summarized all these experimental procedures into a very comprehensive protocol that was published last year. So research groups can follow the step-by-step procedure to apply the system to engineering a variety of different kinds of experimental models. In addition, we built a computational tool which is accessible at tools.genome-engineering.org, where researchers can paste in a region of the genome that they would like to target, and then the tool will computationally try to suggest target sequences either for the wild-type Cas9 or for double nicking to target that region. So toward the end of last year, 2013, we described the study where we try to expand the way that we can use CRISPR to target genes across the genome. And this was done by developing a screening methodology that we call GECKO. It stands for Genome-scale CRISPR knockout screen. The way it works is very similar to a pooled sgRNA functional screen. We first designed single guide RNAs targeting the early and conserved exons of every annotated gene within the human genome, and then using oligo-array DNA synthesis, we can generate specific DNA oligos targeting each one of the sites. And by cloning these oligos into a lentivirus library, we now have a lenti-CRISPR library that can target every single gene within the human genome. So you can transduce a population of cells and then generate a library of cells that collectively have individual genes knocked out. And by applying a screening or bioassay to these cells, we can then identify particular gene mutations that lead to a given phenotype. To first see whether or not the lenti-CRISPR system can work as well as sgRNA to mediate gene knockdown, we compared CRISPR versus sgRNA using a GFP reporter cell line. And what we found is that when the GFP cell line received lenti-CRISPR, we were able to completely deplete GFP expression, whereas for sgRNA, we were able to significantly reduce the level of GFP expression, but we weren't able to completely deplete. And so given this result, we started to apply this to a model of a melanoma. So we took the human A375 cells and then we transduced them with the lenti-CRISPR library so that we'll have a library of melanoma cells with the V600E mutation that have individual genes knocked out. And by applying a drug called vemurafenib onto these cells, what we found is that we were able to identify specific genes when knocked out is able to mediate a resistance against the vemurafenib drug. And by comparing this screen with the sgRNA pooled screen, we found that CRISPR can actually work quite well. For the top 100 targets, we found that consistently the top 100 gecko targets had a lower p-value, suggesting higher significance than the sgRNA hits. And if we zoom in a little bit and look at the top 10 targets, we find that the degree of consistency across the multiple guide RNAs targeting the same gene is much higher than the multiple sgRNAs targeting the same gene. So there's more agreement between the different CRISPR sgRNAs than the different sgRNAs. And functionally, the ability of CRISPR to mediate a complete gene depletion versus sgRNA mediating a gene knockdown can also have a functional significance. So by applying the vemurafenib drug to A375 cells that have the NF2 gene knocked out, we found that when you have CRISPR knockout, we see a robust growth phenotype with this NF2 mutation. But when we do sgRNA knockdown, we don't actually see that. So in cases where only complete depletion of a gene can provide a biological readout, CRISPR may likely have an advantage. In addition to making genetic changes on the genome, CRISPR can also be used to modulate transcription or recruit effector domains to specific locations on the genome. By mutating both of the catalytic domains on Cas9, we can convert it into dCas9 so that we can recruit an effector domain to the target. And the effector domain can be a transcription activator or repressor or some kind of epigenetic modifying enzyme or even GFP to visualize a specific genomic locus. So what we found is that indeed this system can work. So by fusing Cas9 to VP64, we can directly activate an endogenous gene. And by fusing Cas9 to SID4X, we can then repress an endogenous gene within the human genome. So that's all I want to say today. In summary, the CRISPR and also the TAIL systems can provide an easy and robust way to develop genome engineering and epigenome engineering reagents to study both in animal and also cellular models for a variety of basic biological questions and disease processes. All of our reagents described here have been deposited at Gene, so it's readily accessible to the community. So finally, I just want to acknowledge our wonderful collaborators, Luciano Mirafini, David Altschuler's lab, Phil Sharp's lab, the RNAi platform at the Broad Institute, Radolphe and Yi Zhang at MIT, Whitehead, and also Harvard, respectively. And then also all of the funding agencies, the NIH, the NIMH, the different private foundations like Cat Gates, McKnight, so forth, who have provided funding to make this work possible. Thank you very much. Back to you, John.
And thank you, Feng, for your wonderful insights on genome editing using CRISPR-Cas9. We've learned a lot about CRISPRs from you and Radolphe, so thank you. Again, if you want to send a question to our panelists, please use the Ask a Question box on the lower left of your console and then hit Submit. George Church is eagerly standing by and ready to continue our discussion. So, George, you're up.
Thank you, John. This is George Church from Harvard, and I'm going to begin with my thank you slide and thank the previous two speakers as well. This is also my conflict of interest slide, and we're going to be focusing on applications, going beyond what the two speakers talk about to exactly how we're going to impact medicine and other systems. Now, we and others have explored all 7 of these DNA targeting systems over the year. CRISPR is the star today, but one of them is where DNA does the recognition in the genome, 2 where RNA does the recognition, and 4 where proteins do the recognition. The really powerful way using RNA is that you can just program 20 base pairs and move on from there, and so you can make hundreds of thousands of these. So just a quick review, 1987 we saw this in prokaryotes and later in archaebacteria. It wasn't until 2013 we saw them in really applied to eukaryotes, and now over 16 different organisms, basically every organism that's been attempted can use these to cleave or epigenetically modify cells. So that we're going to see these applications now that I'm focusing on today. Reviewing what Radolphe and Feng said, Cas9 is large, but our lab and probably others have developed smaller orthologs, and these fit fine into current vectors. Doesn't seem to be a limit. Particularly the PAM limitations, that is to say the protein components, have already been expanded to many orthologs, and the number grows every day of well-characterized new PAMs. So that's not a big limitation. Feng mentioned the off-target cleavage being greatly improved, maybe 1,000-fold by pair nicases, and he also mentioned the shareability of the SGRNA gene. I'd like to take a moment to talk about the challenges of sharing human data in the context of the CRISPR, so we can enable pre-competitive technology sharing and data sharing. One that's been anointed now by the National Institute of Standard Technology and the Food and Drug Administration for these human genome standards, and one that we use extensively for testing the CRISPR-Cas9 for human gene therapy and so forth, are these personal genomes cellular components. This is the genome in a bottle, is the genome standards, and they're starting with 8 trios. We feel that this is a major component of precision medicine, is merging the whole genome sequencing and the next-gen editing of genomes, such as CRISPR. This cohort has extensive functional data, environmental data, and it's the world's only open access data set. It's now international, many international centers. And we have these cell lines as fibroblasts or as IPS with Cas9 already in it, and it's a very easy way to test out potential therapies, not only gene therapies, but more conventional therapies. And if you'd like to join us and learn more about that, there's an annual meeting this April 29th and 30th, and these are actually the participants who donated cells to this sort of effort. So now, CRISPR. We've talked about tools. We've talked about Feng mentioned libraries at the end, disease models that we can make in mouse and primate and so forth. But the next step is deciding what we're going to aim this powerful technology at. We can some diseases can be prevented by proper gene counseling, and so there's a subset of these which are more appropriately dealt with by gene therapy, and we'll talk about that a little bit. There's soma versus germline. The ultimate in preventative medicine would be germline. but I think we're going to be seeing mainly soma until the safety and efficacy is unambiguously proven for many diseases. We can fix rare diseases, and I think that will be some of the early applications, but there's some interesting common allele applications. I think in the long term that will be the killer app, because gene counseling will deal with so many of the rare diseases. We can, currently there are about 2,000 gene therapies mostly involve adding a gene that's missing, but CRISPR greatly enables deletion and alteration. We can have low levels of multiplexing, basically changing one place in the genome, or we can have high levels. And why we do that at high levels is an interesting question. And two examples as to what your appetite for high-level multiplexing is developing organs for transplantation, either in changing human or animal systems. And then there's ecosystem engineering, which CRISPR can be applied to in a variety of ways. So how do we do multiplexing? What are the limits here? Here's an experiment where we use multiplexing for the epigenetic reprogramming activation that Radolphe alluded to. And here you can put in 10 different guide RNAs simultaneously, and the 10 is much more powerful than any of them individually, getting, in this case, 35-fold induction. And another paper that used 73 guide RNAs simultaneously, not necessarily proven to be all at once, but we're getting sort of into the range where we don't really know where the upper limit is for this sort of multiplexing. So one of the things that's incredibly enabling about CRISPR is this ability to make dozens, hundreds of thousands of guide RNAs and test either in low throughput or high throughput hypotheses. So the hypotheses flowing out of genome sequencing and other association studies can now be tested on very small cohorts. False positives are no longer as daunting as they used to be. And so N-of-1 studies where we move from correlation, which you might get in large cohorts, to causality, which can be tested in small ones. The tests can be either in animals or in humans, human cells, human organoids. And here's an example of one where you have very, very rare traits, not necessarily diseases, where you have a myostatin double null in humans, or a myostatin receptor double null in these two young individuals on the left, and then reproduced in 3 different animal models, cows, dogs, and mice. And that's causality. You could also do it in humans. If for some reason or other the animal model is not appropriate, is missing that anatomical structure or physiology, you can do it in human organoids, and these are getting increasingly sophisticated and increasingly well vetted for their relationship to natural human organs. I urge you to take a look at this review article, and we've had a wonderful interaction with our colleagues at the Wyss Institute for Biologically Inspired Engineering, and Don Ingber and Kit Parker have been working on, for example, the lungs and the cardiac models, and we can introduce with CRISPR human mutations either from the literature, databases, or and ask whether a particular position out of the millions of 3 million that might differentiate between 2 people, you can introduce one at a time and see whether a particular base pair or small region is necessary and sufficient for physiological impact at the molecular, cellular, and tissue level. For example, contractility of heart tissue, or physiology of lung ownership, and so on. So, how do we make this decision as to which genes are the appropriate ones? We have, if we have gene therapy, do we apply it to genes that are currently handled by genetic counseling, such as Tay-Sachs, which has been almost eliminated via gene therapy, by gene counseling? So there are now over 1,970 gene therapy clinical trials, whether they're viral or non-viral delivery, mostly adding genes in phase 1, 2, and 3 clinical trial. There's one that's already emerged, Glybera, licensed or approved in Europe. But many of these are curing these rare diseases, which is becoming increasingly possible to detect with carrier testing, not of one gene at a time based on your family history, but dozens at a time, and very soon the whole genome at a cost that is embedded in your healthcare. So here's an example of a company, Good Start Genetics, and they have a panel that can be suggested by ethnicity or by family, or can be just done preventatively in general. But you can see there's a large number of these that can be done simultaneously. So, and that brings us to those are mostly rare, and they can be detected in the potential father and mother, and avoided. But if the father and mother share allele, they have no way out, or let's say the entire human race for the most part shares alleles, we have a larger market and a more serious problem. And there are examples of these variations, which we'll call rare protective alleles, rather than the rare deleterious ones. These are the rare protective alleles. And we have some examples here. The myostatin I already mentioned, LRP5, results in extremely strong bones, the opposite of osteoporosis. And this is a particular allele in a heterozygous state. Example here, glycine 171 to valine. PCSK9 is really exciting. You can lower coronary artery disease in heterozygous and homozygous. You lower LDL cholesterol to unbelievably low levels, and people are quite healthy. CCR5, CXCR4, and FUT2, all in their double null state, remove the receptor from the surface of the cells that would be the virus receptor for either HIV-1 or norovirus. And finally, APP, this particular allele, A673T, is not predisposing to Alzheimer's in rare deleterious sense, but is actually protective. Now, what do we do with these rare protective alleles? Almost nobody has them, and we'd like to get them out to the world. These various gene editing, gene targeting methods that we've been taught, that I mentioned in my very first slide, one of them is already in phase 2 clinical trials from Sangamo. This is a zinc finger nuclease, but it's illustrative of what's riding right behind them with the CRISPR. And in this case, you'll make a double-strand break where the protein determines where in the genome the needle in the haystack, 1 in 3 billion base pairs, to cleave, and you make a mess with non-homologous end joining, as Feng has described. You basically insert or delete a small number of base pairs, highly variable, but enough to make a null. In this case, CCR5, and a double null will mean that the T cells from that patient, his own or her own T cells taken out and put back in, will make them, or put into the precursor cells, will make them resistant to the HIV, even if they have full-blown AIDS at the time. This is a huge breakthrough, not only because you're removing DNA, which is not what most gene therapies have done in the past, you're removing, but you're also treating something that's very common. Essentially everybody on the planet, with a very small number of exceptions, has a functional CCR5 gene or two. Now, to generalize this a bit more, and to illustrate the difference between non-homologous end joining, where you make a single cut and essentially remove the functionality of that gene, you can make two cuts and you get a clean deletion, and it tends to be less of a mess. It's exactly what you want. It tends to be precise. Or you can use two cuts with two guide RNAs and hence two CRISPR complexes, and bring in a donor DNA and do the homologous recombination. Here, Susan Byrne and colleagues are illustrating this by changing human cell surface for protein Phi1 with a mouse equivalent, mouse Phi1. this is done by FACS analysis. We have great fluorescent antibody reagents for both the human Thywan and the mouse Thywan. And so you can see this transition from the human Thywan in the lower right quadrant of each of these 4 FACSs going from left to right. You've got one with just the left guide RNA, one with just the right guide RNA, one with both, and then the control with no guide RNAs. And you can see in that no guide RNA on the far right, everything's in the human quadrant, and then with both the left and the right, you have a high percentage that has been converted to mouse Thywan, essentially without selection. But we're using FACS here to do quantitation. So in summary, we've first 2 speakers introduced us to these wonderful, the history of CRISPR, the tools, the libraries that one can make, hundreds of thousands, interesting disease models in animals, and then we're moving this into gene therapy. Feng and I and others have founded a company called Editas, but there will be many opportunities here, and there will be an interesting discussion of just as we determine whether it is very safe and very effective in somatic gene therapy. There will be a discussion of further preventative medicine, and we will have the opportunity doing common alleles, treating problems that are common to most humans, or maybe all of us, like aging and dying. And finally, just to emphasize, the real power here is we can delete and alter, and that we can try out a large number, either simultaneously or iteratively. So thank you very much. Back to John.
And George, thank you. I'm not exaggerating when I say your talk, as well as those of your 2 colleagues, was a veritable tour de force on CRISPR. So thank you all. Before we begin our Q&A session, I'd like to ask you to disable your pop-up blockers. A short survey on this webinar will be appearing in a moment, and very much appreciate your feedback on this webinar. Okay, so let's take a question from the audience.
Rudolph, we have an interesting question for you. Does Cas9 cleavage also leave cohesive ends?
Thus far, in native Cas9 systems that have been biochemically characterized, published and unpublished, it does appear as though all cleavage generates double-stranded DNA breaks blunt. But if you're interested in having overhangs, one option would be to use the nickase variant in combination with overlapping spacers to target the 2 different strands.
Okay, thank you. Feng, a question for you. How can we use this method to identify the function of different genes? In other words, what are the uses of CRISPRs in functional genomics? Feng? Did we lose you, Feng?
Hello, I'm here. Sorry, I think my speaker was on mute.
Okay, so do you have that question? How can we use this method to identify the function of different genes? In other words, what are the uses of CRISPRs in functional genomics?
So CRISPR can be used to achieve efficient gene knockout in different kinds of functional experiments. For example, one of the recent studies that we reported, we can generate a library of CRISPRs targeting almost every single gene in the genome. And then by applying this library of CRISPR in an analogous fashion as sgRNA libraries, we can knock out individually different genes and then apply selection pressure to see what are the functions of specific genes in a given biological output. So for example, you can identify mutations that confer drug resistance to different kinds of cancer drugs.
Thank you, Feng. And George, will offset targeting strategies decrease the recombinant efficiency?
So the offset targeting or double nickase strategy does not appear to impact recombinant efficiency. In fact, there's a late breaking news from Keith Young's lab that if you reduce the guide RNA by a couple of nucleotides at the 5' end and add that to the double nickases, you can bring the the targeting efficiency, the off-targeting down by a factor of 5,000, and none of those has an impact on recombinant efficiency.
Okay, thank you. And Rudolph, for homologous recombination, what's the maximum insertion length? Also, is DS out at 2 sites better than DS out at 1 site?
To the best of my knowledge, I think this is yet to be determined whether there's a limit as to how large the templates can be for insertions. But there's some preliminary evidence that it can be several hundred base pairs, maybe even several kbs. But we'll have to wait a little bit for that. With regards to the other part of the question, I mean, I think having 2 cuts is an advantage in terms of increasing selection and pressure and cleavage efficiency, but it will likely come at the cost of survival of the cells and then efficiency of recombinations.
Okay, thank you. And Feng, here's a very interesting question. How does the CRISPR system avoid attacking the host bacterial genome? And this pertains to controlling off-target cleavage events.
The way that CRISPR avoids cutting the native bacterial genome is by the recognition or the requirement of a PAM sequence. In the CRISPR locus, even though the target sequence is present, that sequence is not flanked by the protospacer adjacent motif, the PAM sequence. And so that's how the enzyme avoids cutting it.
Okay, great. And George, is there a way to select between NHEJ and HDR for further genetic modification after the introduction of the double-strand break?
Well, so first of all, you can bias HDR versus NHEJ by not making a double-strand break. By putting in a single nick, you heavily bias it towards recombination in many of the systems that have been characterized. It's not necessarily completely general. You will reduce the efficiency, the overall efficiency, but the ratio changes considerably. Furthermore, if you put 2 double-strand breaks, you'll get a clean deletion with generally with none of the collateral damage you get with non-homologous enjoining.
Okay. And Rudolph, how are insertions made?
Essentially, insertions are generated by the design of the template you use for homology-directed recombination, HDR or HR. And what you have to do is in your template sequence you provide as a cure for the cleavage repair, you have to essentially separate the spacer sequence and especially the cleavage part at the 3' end, and on one side, and then on the other side you would have to put the PAM in the repair template. So you have to separate the PAM from the spacer and insert your target there.
Okay, and we have a question. I'm going to direct it first to Dr. Feng, and then George can take the same question. Is it possible for a new array to have homology with genomic DNA, which would cause cutting events within the bacterial genome? Feng first.
Sorry, could you maybe restate the question?
Sure, yeah. It's sometimes I have to try to interpret these. Is it possible for a new array to have homology with genomic DNA, which would cause the cutting events within the bacterial genome? Not quite sure what that means, but I'll give it a go.
So sure. So the CRISPR arrays are assembled by having concatenated repeat sequences interspaced by the target sequence. The requirement for cleavage of a target sequence is that it has to be flanked by the right PAM sequence. And usually the natural CRISPR system avoids self-cleavage by not choosing target sites that are similar to the repeat sequence, at least on the 3' end. And so it depends on what the array that has been generated. As long as the individual recognition sites don't have the PAM sequence, then it should not be cut.
George, can you take that too?
I think Feng did a great job.
Okay. Then let's move over to Rudolph, and let me get down to this question. Rudolph, what is the difference between RUIC plus HNH, looks like plus, and it's not coming out clear, RUIC and HNH+. Do they not result in single-strand cuts?
Yeah, so I'm assuming the question pertains to the variant functional forms of Cas9 with RUVC plus or minus in combination with HNH plus or minus active domains. And essentially both variants would be single-strand DNA nicases, but the strand that would be nicked by the RUVC plus versus the strand nicked by the HNH plus would be on opposite sides of the DNA helix. So whether you want to nick on the plus strand or specifically on the minus strand, you would have to use the variant accordingly. So it's a question of orientation on your helix.
Okay, thank you. And Feng, which CRISPR-Cas system will be efficient to knock down long non-coding RNAs?
There are different strategies of knocking down a gene. For non-coding genes, you may choose to introduce two different guide RNAs that flank the non-coding RNA of interest, and Cas9 will be able to excise out the non-coding RNA.
Okay, and George, question for you. Is it feasible to correct large, greater than 5 kilobyte, deletions, losses, or duplications, gains?
So with homologous recombination, you can insert. We don't know what the upper limit for insertion is, but it's probably similar to previous homologous recombination, and probably quite large with, say, bacterial artificial chromosome 100 kilobase type of substrates. The largest I've heard of for deletions is on the order of 10 megabases, so you can really have very large deletions, which would cover. Which you could use that to remove duplications. The limits would, of course, be if you have highly repetitive sequences of getting just the right number of repeats out.
Thank you very much. Well, unfortunately, we've run out of time, but please note that this webinar will be archived for 6 months on our website, www.genangnews.com. If you missed parts of it, you can watch it again, or you can recommend it to your friends and colleagues, which we highly recommend. Also, we had quite a lot of questions in this webinar, and the panelists will try to answer as many as they can. Hopefully they'll be able to get to some of these. Hey, I want to thank the panel again for the outstanding presentations, and to say thank you to our audience for your attention and for your very, very thoughtful questions about various topics brought up during the webinar. And thank you to our sponsor, OriGene. Bye for now.